Hello, I'm Joshua Shulman. I'm an assistant professor of neurology, molecular and human genetics, and neuroscience at Baylor College of Medicine, and an investigator in the Duncan Neurological Research Institute at Texas Children's Hospital. Well, as a neurologist, I'm expert in disorders that affect the brain. My focus is on adult neurological disorders, and in particular, Alzheimer's disease and Parkinson's disease, which are neurodegenerative disorders. Alzheimer's disease is incredibly common. It affects more than 5 million people in the United States and 20 million people worldwide. It's a devastating problem, takes away people's ability to think and remember, and unfortunately, because it's currently incurable and we have no way of preventing its progression, there's a huge and urgent challenge to have better ways to treat this condition. We know that outside of brain cells, certain protein aggregates called amyloid plaques form, and these aggregate proteins have toxic influences on the function of neurons and ultimately might lead to their demise. In addition, there's another kind of protein aggregate called a tau tangle that forms within the neurons of the brain. And it's these two pathologies that accumulate over time and we think lead to the dysfunction of the brain and the loss of a large number of brain cells. So what we're trying to understand is how certain genetic variations might interact to affect the risk of disease and then its progression in the population. The goal of the research is that by understanding something about genetic risk, we may be able to intervene and alter those early mechanisms, which would lead to either a prevention or a cure. But we take two broad approaches. On the one hand, we participate in new gene discoveries, and these are studies that take place in humans. So we've participated in studies with tens of thousands of subjects who have Alzheimer's disease to try to improve our power to identify the types of changes that probably have epidemiologically important effects across the population. Once you find a gene or a group of genes that have a role, of course, it's quite limited what you can do in humans experimentally. Usually, to understand mechanism, you need to go to some experimental system. And that's where animal models, including the fruit fly or the mouse, come into play in the laboratory. We have too many gene candidates. Fruit flies have been particularly useful for the work that we've done over many years. It takes a lot of time to understand the genetic impact of manipulations at any single gene. So in order to try to look at a large number of genes at the same time, using a simple model system like the fly has real advantages. Flies actually have brains. They have some of the same chemical messengers that human brains have. Flies, the surprise of many, actually have fairly complex behaviors. Although the fly is simple, that simplicity actually becomes a valuable tool for us. We're focused on a group of genes that we think have an important role in synaptic biology. And the synapse is the structure within the brain by which one cell communicates to another. Synapses are thought to become dysfunctional and degrade at a very early stage in Alzheimer's disease. It's one of the earlier changes that we can detect in the brain using certain imaging tools that we can even recognize disease before, say, a neurologist would diagnose somebody with Alzheimer's disease. Now, when we identify something in the fruit fly that is suggestive of a genetic mechanism that might be important in Alzheimer's disease, ultimately, the goal is to translate those lessons back to human patients and the human population. We're at an exciting time in Alzheimer's disease research. There's increasing recognition that the disease starts many, many years before patients first present with symptoms. We have new tools to detect those changes before people actually have trouble with their memory. There are now exciting ways of imaging Alzheimer's disease-related changes in living subjects. And so these types of approaches give us hope that people who are at the earliest stages developing this condition could be treated with drugs that might alter or prevent the disease from progressing.